Hello viewers. Today I will once again discuss on protein metabolism but a different aspect. I will start off with how the different amino acids enter the TCA cycle following different routes. Now there are 20 different naturally occurring amino acids with different carbon skeletons. So it is very obvious that these amino acids, when they are converted to TCA cycle or their precursors, follow diverse pathways. Now here in this particular slide, we can see that how the different amino acids enter the TCA cycle following different routes. Now based on these catabolic pathways, amino acids can be classified into three major groups. These are glucogenic, ketogenic and both glucogenic and ketogenic amino acids. Now what are glucogenic amino acids? Glucogenic amino acids are those amino acids whose carbon skeletons can be converted to either pyruvate, fumarate, oxaloacetate, succinyl-CoA and alpha-ketoglutarate. All these are glucose precursors and hence these amino acids are called glucogenic amino acids. Now 13 amino acids fall under this category and the examples include glycine, serine, alanine, methionine, isoleucine, etc. Now first we will have a look that how the different glucogenic amino acids are converted to different TCA cycle intermediates. In the first diagram, we can see that alanine, glycine, serine, threonine and cysteine, all these are converted first to pyruvate and these then enter the TCA cycle via acetyl-CoA. However, two amino acids like asparagine and aspartate are converted to oxaloacetate, which then enters the TCA cycle. Even 5 another amino acids like histidine, proline, glutamine, glutamate and arginine are converted to alpha-ketoglutarate, which is another important TCA cycle intermediate. 3 amino acids like isoleucine, methionine and valine are converted to succinyl-CoA, which then enters the TCA cycle. So you can get an idea that how these different glucogenic amino acids enter the TCA cycle following different routes. Now I will discuss what are ketogenic amino acids. Now ketogenic amino acids are those amino acids whose carbon skeletons can be converted to either fatty acids or ketone bodies. 2 amino acids fall under this category, which are lysine and leucine. Now here in this particular diagram we can see that how leucine follows a multi-step pathway and ultimately it is converted to acetoacetate, which is a ketone body. Again, lysine follows a multi-step pathway and it is ultimately converted to acetoacetate, which is a ketone body. So both these amino acids generates ketone bodies and hence these are called ketogenic amino acids. The last group includes glucogenic and ketogenic amino acids. As the name suggests, these amino acids can be converted to both carbohydrates and ketone bodies. Important examples include the aromatic amino acids tyrosine, tryptophan and phenylalanine and also isoleucine and threonine. Now as we can see in this particular slide that tryptophan is converted to alanine, which is a glucose precursor, and also to acetoacetate, which is a ketone body. So it generates both glucose precursor and ketone body, hence tryptophan is both ketogenic and glucogenic amino acid. Similarly, phenylalanine and tyrosine are degraded to fumarate and acetoacetate. Fumarate is an important TCA cycle intermediate and also a glucose precursor, whereas acetoacetate is a ketone body. So these two amino acids are also both glucogenic and ketogenic amino acids. Now after giving you a brief overview of how the 20 different amino acids enter the TCA cycle following different routes, I will now discuss in detail that how the amino groups are removed from the amino acids in three major ways. In this particular slide, we can see that there are three major nitrogenous excretory products, which are ammonium ion, uric acid, and urea. Based on this, living organisms can be classified into three major groups, which are ureotelic, ammonotelic, and uricotelic organisms. Now I will discuss what are ureotelic organisms. Living organisms which excrete the excess amino nitrogen as urea are called ureotelic organisms. Examples include terrestrial vertebrates, adult amphibians, and sharks. The next group include ammonotelic organisms. These are organisms which excrete the excess amino nitrogen in the form of ammonium ion. Most aquatic animals like bony fishes and aquatic vertebrates excrete amino nitrogen in this way. The last group includes uricotelic organisms, and these organisms excrete excess amino nitrogen in the form of uric acid. Now, uric acid is the least toxic and least soluble in water amongst the three nitrogenous excretory products. Uricotelic organisms lack two major urea cycle enzymes, which are carbamoyl phosphate synthetase 1 and arginase. So urea cannot be generated by these organisms, hence they excrete excess amino nitrogen in the form of uric acid. Examples include birds, land-dwelling reptiles, and amphibian larvae. Now, accumulation of excess ammonium ion in blood causes hyperammonemia. The problems associated with hyperammonemia are as follows: changes in cellular pH, increase in reductive amination, increase in L-glutamine synthesis, increase in L-glutamine outflow from brain, and also increased gluconeogenesis, thereby increasing the ammonia load further. Now first I will discuss in detail each of the problems. In first step, we have seen that the problem associated with hyperammonemia is changes in cellular pH. Now ammonium ion, which is the protonated form of ammonia, is a weak acid, whereas ammonia is its conjugate base. Now most of the reactions excrete amino nitrogen in the form of ammonium ion, and whereas some of the reactions, as those catalyzed by adenosine deaminase, release ammonia directly. So this may cause alkalization of the cellular fluid, which may have complex effects on cellular metabolism. The second problem is increase in reductive amination. So we can see here in this slide the example of increase in reductive amination. What happens is that alpha-ketoglutarate is converted to L-glutamate. The enzyme catalyzing this reaction is L-glutamate dehydrogenase, requiring the coenzyme either NADPH or NADH, and this results in reduction in the mitochondrial pool of alpha-ketoglutarate as also the coenzymes, and as a result the TCA cycle is depressed, thereby resulting in concomitant loss in ATP production. The next problem associated with hyperammonemia is increased L-glutamine synthesis. It is harmful for the brain and have adverse effects on cerebrospinal fluid. Now, as we can see in this reaction that from L-glutamate, L-glutamine is synthesized. The enzyme is L-glutamine synthetase, and this results in decrease in brain's pool of L-glutamate, which is an important neurotransmitter, and thereby results in decrease in the production of another inhibitory neurotransmitter, which is gamma aminobutyric acid. Now, L-glutamine synthesis also depletes ATP. Increased L-glutamine synthesis also causes L-glutamine outflow from the brain, and as a result, tryptophan comes in through the same antiporter system and may cause the generation of another neurotransmitter called serotonin. And lastly, the problem associated with hyperammonemia is increase gluconeogenesis. Now, excess ammonium ion stimulates glucagon secretion from pancreatic alpha cells and thereby results in increased gluconeogenesis and increasing the ammonia load further. Excess ammonia may cause excretion of urea through urine, and other symptoms include nausea, dizziness, blurring of vision, slurring of speech, ataxia, mental retardation, lethargy, coma, and ultimately death. Now, what is urea cycle? Urea cycle or ornithine cycle or Krebs-Henseleit cycle was first discovered by Sir H. A. Krebs and K. Henseleit in 1932. Later on, the detailed enzymatic steps was discovered by S. Ratner and P. P. Cohen. Now I will discuss the details of urea cycle. Here in this particular diagram we can see that urea cycle consists of 5 major steps, and this is one molecule of urea, and the 2 nitrogen atoms and the carbon atoms come from ammonium ion, aspartate and bicarbonate respectively. Now, one of the interesting feature of urea cycle is that it is compartmentalized. As we can see in the diagram that the first 2 steps of urea cycle occur in mitochondrial matrix, whereas the last 3 steps occur in cytosol. Now I will tell you what is the need for this compartmentalization. Fumarate, one of the important TCA cycle intermediate, is present in mitochondrial matrix, which inhibits the enzyme argininosuccinate lyase. And also arginine is present in mitochondrial matrix, which also inhibits the same enzyme. So urea cycle must be compartmentalized so that urea formation can take place. Now I will discuss in detail how the ureotelic organisms excrete the excess amino nitrogen via urea cycle. I will tell you the details of each of the 5 steps of urea cycle. Now in step 1, as we can see in the diagram, results in the formation of carbamoyl phosphate from ammonia and carbon dioxide requiring 2 ATP molecules, and the reaction is catalyzed by carbamoyl phosphate synthase 1. Now eukaryotes uses another counterpart of carbamoyl phosphate synthetase, that is CPS2, which is cytosolic and uses glutamine as the nitrogen donor for the synthesis of pyrimidine. In step 2, the enzyme catalyzing the reaction is ornithine transcarbamylase, and it is also a mitochondrial enzyme. The carbamoyl group from the carbamoyl phosphate reacts with ornithine to form citrulline. Citrulline, after its formation, enters the cytosol where occurs the 3rd step of urea cycle. The urido group of citrulline reacts with amino group of aspartate to produce argininosuccinic acid. The enzyme catalyzing this reaction is argininosuccinate synthetase. This reaction also requires ATP, and the pyrophosphate in this reaction is hydrolyzed by pyrophosphatase to produce inorganic phosphate, and as a result the overall reaction is driven in the forward direction. In step 4, from argininosuccinate, arginine and fumarate are produced. The enzyme is argininosuccinate lyase or argininosuccinase. And in the last step of urea cycle, there occurs formation of ornithine and urea from arginine. The enzyme catalyzing the reaction is arginase. Ornithine, after its formation, enters the mitochondrial matrix to begin another round of urea cycle. There is an interesting information that human beings excrete about 22 pounds of urea per year. The overall reaction of urea cycle is shown here. It is the interaction of ammonium ion, carbon dioxide, ATP, aspartate and water molecule to produce urea, ADP, AMP, inorganic phosphate, pyrophosphate and fumarate. Now if we have a look into the energy cost of urea cycle, we can see that 3 ATP molecules are consumed in urea cycle, whereas fumarate after its production in urea cycle enters the TCA cycle to produce 3 molecules of ATP. So there is no extra drainage of energy and the net expenditure of energy reduces in urea cycle. Now I will discuss how urea cycle can be regulated. The first way of regulation is through allosteric activation. The allosteric activator is N-acetyl glutamate, which activates the enzyme required in first step of urea cycle, which is carbamoyl phosphate synthetase 1. Now this is a very important step of urea cycle and we may call it the rate limiting step, as because all the other steps are dependent on this step only. N-acetyl glutamate is formed from glutamate and acetyl-CoA. The second way of regulation is through enzyme induction. Now it may occur in 2 cases. One is in case of protein-rich diet, another is in case of starvation. Now if we have a protein-rich diet, the dietary proteins undergo hydrolysis to free amino acids, which then generates excess ammonia, thereby stimulating the urea cycle enzymes. The second case is in case of starvation. In case of starvation, muscle proteins undergo hydrolysis to generate free amino acids, then generate excess ammonia, thereby inducing the urea cycle enzymes. The third way of regulation is through urea cycle intermediates. Now increase in concentration of any one of the urea cycle intermediate may cause inactivation of the preceding step enzymes. Krebs bicycle, which is nothing but interlinkage of TCA cycle and urea cycle. So in this particular diagram we can see that how the 2 cycles are interlinked. Let me explain it to you. Fumarate after its production in urea cycle is converted to oxaloacetate via malate. Now this oxaloacetate may have several possible fates. It may be converted to pyruvate, it may be converted to aspartate, it may condense with acetyl-CoA to produce citrate, or it may produce glucose through gluconeogenic pathway. Now aspartate is an important nitrogen donor of urea. I have already told this. So this aspartate comes from oxaloacetate and it, after being formed in mitochondrial matrix, enters the urea cycle to participate in it. Alpha ketoglutarate is formed here as because the reactions of TCA cycle and urea cycle are intertwined together. So this is the so-called Krebs bicycle. There are other excretory nitrogenous products besides ammonium ion, urea and uric acid. For example, spiders excrete amino nitrogen in the form of guanine instead of uric acid. Many fishes excrete nitrogen as trimethylamine oxide. In higher plants, glutamine and asparagine function in the storage and transport of amino groups. Now lastly, I will discuss on various genetic defects of urea cycle enzymes as also various inborn errors of amino acid metabolism. Each of the enzyme required in 5 steps of urea cycle are very important and their deficiency may have serious consequences. As we can see that the first enzyme CPS1, its deficiency leads to hyperammonemia type 1 and the symptoms are already shown in the chart. Some of them are protein intolerance, convulsion, ataxia, etc. The second enzyme is OTCase or ornithine transcarbamylase and its deficiency results in hyperammonemia type 2 and the symptoms include ornithine and glutamine increase in concentration, ammonia toxicity symptoms like protein intolerance, etc. The third enzyme, which is argininosuccinate synthetase, if deficient, leads to citrullinemia, that is increase in citrulline concentration in blood and urine, hyperammonemia and mental retardation. The fourth enzyme is argininosuccinate lyase, and it, if deficient, results in argininosuccinic aciduria. The symptoms include argininosuccinate level increase in blood, urine and cerebrospinal fluid, vomiting, mental retardation, etc. And the last enzyme required in urea cycle is arginase, whose deficiency causes hyperargininemia. The symptoms include increase in arginine level in blood, cerebrospinal fluid and brain, and urinary levels of cysteine, lysine, ornithine, arginine increase, etc. It is already shown in the chart that how the deficiency of different enzyme leads to different types of inborn errors. Some of the important ones are albinism, which results in prevention of melanin synthesis from tyrosine. The enzyme deficient is tyrosine 3 monooxygenase, and it results in lack of pigmentation, white hair, pink skin. Next inborn error of metabolism is alkaptonuria, and the enzyme deficient is homogentisate 1,2-dioxygenase. The symptom is dark pigmentation in urine. The third inborn error of metabolism and one of the important inborn errors is phenylketonuria. As the name suggests that it prevents the conversion of phenylalanine to tyrosine, and the enzyme deficient is phenylalanine hydroxylase. The symptoms include neonatal vomiting, mental retardation, etc. Other inborn errors of metabolism includes argininemia, argininosuccinic aciduria, maple syrup urine disease, and many others. I have given you a detailed discussion on how the 20 different amino acids enter the TCA cycle via different routes, as also I have discussed urea cycle in detail and the genetic defects of urea cycle enzymes and different inborn errors of metabolism, amino acid metabolism. Thank you. This is all for today's lecture.